Hello, my name is Wes Gherman, and I'm a second-year medical student at the University of Toledo. As part of Dr. Giovannucci's lab, I will be presenting on otolith N organs hair cell polarity-specific segregation of primary vestibular afferent fibers into the brainstem and cerebellum in chick embryo. First, let's briefly introduce the topic of interest. In all vertebrae, the otolith N organ's hair cell morphological polarity is reversed across a line known as striola. The function of this polarity reversal at the striola is currently unknown. For this study, we investigated whether the central projection of the different polarity domains in the otolith N organs occur in other classes in vertebrae such as aves. For materials and methods, lipophilic retrograde tracers including NeuroView Red and NeuroView Maroon were implanted in the vestibular nuclei in the nodulus and uvula of the cerebellum respectively in post-fixed pre-hatching chick embryo. After the diffusion time was complete, the macula of otolith N organs were dissected out and examined with confocal microscope. First, a brief overview of the results. In the macula of the utricle, NeuroView Red placed in the vestibular nuclei retrogradely labeled a distinct domain of hair cell located inside the striola region, whereas the NeuroView Maroon retrogradely labeled hair cells located outside the striola. Similar findings were observed in the macula of the saccule. Here in Figure 1, you're looking at the utricular macula, which is responsible for the detection of linear acceleration in the horizontal plane. Looking at images A and B, you can see rather clearly that there is a line differentiating the two polarity domains. This line is what we refer to as the striola. The polarity domain within the striola in red is projecting to the brainstem, whereas the polarity domain outside the striola in green is projecting to the cerebellum. Images C and D are higher magnification images of a nerve terminal and hair cells showing the same segregation pattern. Here in Figure 2, you're looking at the saccular macula. Images A and B show labeling for projections going to the cerebellum and vestibular nuclei respectively. Image C is a merged image of A and B, which shows the comparative segregation pattern. Notice how in the saccule it appears that the cerebellar projections are more so located within the striola, whereas brainstem projections are located outside the striola. This contrasts with the utricle, which showed the opposite projections of the polarity domains. Now, Figure 3 is looking at something distinct from mammals. This is a look at the laginar macula, which is something that mammals do not possess. The avian laginar macula is a markedly curved otolithic organ located at the distal tip of the cochlear duct and is known to have projections to the vestibular nuclei and cerebellum. Images A and B show labeling for projections to the cerebellum and brainstem respectively. Image C shows the merged images of A and B. Notice that there's no clear striola observed in the laginar macula as opposed to what is observed in the utricle and saccule. In conclusion, our data suggest that the central segregation of the primary otolith afferents of the different polarity domain reported previously in mammals is also conserved in other classes such as aves. Now, shown here in Figure 4 is a proposed model of the function of the hair cell polarity domain across a striola and their differential central projections. This projected model focuses primarily on the utricular macula. Our proposition is that with a given natural stimulus such as head translation, when the activity arising from the nuclear macula is facilitated, the activity arising from the cerebellar macula will be disfacilitated. This disfacilitated cerebellar macula would result in a decrease in the rate of firing of the mossy fiber input to the granule cells and unipolar brush cells within the cerebellar cortex, decreasing their drive on Purkinje cells. Consequently, the Purkinje cell inhibitory influence over the secondary vestibular neurons will be decreased. Thus, the disinhibitory input of the cerebellar macula is positioned to polysynaptically converge with the facilitated excitatory input from the vestibular nuclear macula on secondary vestibular neurons, producing a vectorially uniform effect. In the same scenario, we propose that the activated vestibular nuclei will have enhanced inhibitory inputs to the contralateral secondary vestibular neurons. Okay, and I just wanted to briefly thank all the wonderful faculty who have helped guide this project. So thank you to Dr. Giovannucci, Dr. McClaw, Dr. Frank, my classmate Cassidy Regent, and Asta, who's the undergraduate student working in our lab. Thank you all so much.